Diabetes is a global health challenge with a growing impact. In Mexico, diabetes is one of the diseases with the highest incidence and mortality, as it is the second largest cause of death. It is estimated that by 2021, there will be 12.4 million people suffering from this comorbidity, type 2 being the most common in people over 60 years. However, it can occur at any age. Unfortunately, diabetes can trigger various pathologies and complications that end up in further deteriorating of patients' health. Diabetes is the leading cause of diabetic nephropathy, DN. Both types of diabetes can lead to any type of kidney disease. It is estimated that approximately 1 in 3 people with diabetes may have chronic kidney disease. Since the main job of the kidney is to filter waste and extra water out of the blood to make urine, it is also to help control blood pressure and make hormones needed to live. When the kidneys are damaged, they can't filter the blood correctly, leading to contaminant and waste buildup, and thus to major health issues. Current diagnostic methods rely on the detection of albumin in urine. However, albumin is a biomarker that is found in more advanced stages of the disease. The lack of early diagnostic tests for diabetes-related kidney damage compounds the public health challenge, resulting in more complex patients' prognosis, increased suffering, and healthcare system strain.
For the early detection of diabetic nephropathy, we need specific biomarkers that we can find in the urine. So we came up with the following 2 molecules that will help us with our mission. The first one is transferrin, which is a protein whose function is to fix and transport iron in the blood. This is a glycoprotein that is mainly produced in liver. If transferrin is found in concentration greater than 2.2 to 8.9 nanograms per milliliter, it can be classified as microalbuminuria. On the other hand, our second protein, neutrophil gelatinase-associated lipocalin, or better called NGAL, is a protein of the innate immune system, which is produced by neutrophils and prevents iron utilization by bacteria. It is found in low concentrations in the liver, prostate, and respiratory tract epithelium. When NGAL is presented at a concentration of 51.1 nanograms per milliliter, it is similarly classified as microalbuminuria.
But how are we going to sense these 2 proteins? For the sensing of the biomarkers NGAL and transferrin, 2 peptides with affinity to our biomarkers were obtained from literature, MPEP and TPEP, respecting the initials of their biomarkers to be sensed. These 2 small amino acid chains were selected for their specificity and their reported affinity. Likewise, to validate their reported properties, a simulation of protein-peptide molecular docking interactions was performed using the HDoc server, and the polar interactions between the peptide chains and their biomarkers were analyzed in PyMol of the 10 best model obtained from HDoc. A maximum confidence score metric of 0.83 for TPEP and 0.91 for MPEP was obtained. At the same time, the interactions compared between the obtained models and the literature enhance the use of these peptides for the sensing of the previously mentioned biomarker.
Our test is designed using FixL and FixJ system from Bradyrosmium japonicum that originally sends oxygen outside the cell. This system is divided by 2 important protein complexes, the first one being a histidine kinase chimerized with the peptide sensible to our biomarkers, which is denominated transmitter domain, or as established by our team with the following code. And the response regulator called receiver domain with the code that enables the transcription of reported genes by uniting to the promoter FixK2 and expressing amylCP, a protein visible to the naked eye when the system is active. But how do we activate the system and how the diagnosis test will occur? First, a biomarker appears, whether it's NGAL or transferrin enters the cell-free system by being exposed to a urine sample. When the protein is near the chimeric protein, FixL, it causes conformational change that is propagated by a linker and reaches the ATPase, and it commands it to break ATP and to add a phosphate to the DHP region. This phosphate, through a phosphotransfer reaction, will reach the rec domain in FixJ and activate the effector domain that will consequently bind to the promoter in the reporter sequence of glucuron protein. Allowing us to know if the test turns blue, it means the system is sensing the biomarkers and allowing the diagnosis of early nephropathy. The test will be divided in 3 sections following the principles of lateral flow tests. One where the chimeric protein with affinity for NGAL will have its system the other one where it will be the transferic chimeric protein, and other one for the chimeric protein with affinity for our control, albumin. All the components and reactants will be dry and activated with the sample. Looking for the feedback of the implementation of our test, we developed some activities to build our human practices.
Join us as we explore the human practices efforts that the iGEM Tec CEM team did. First of all, we started making research to define and know more about the problem that we were trying to solve. We reached out to two distinguished medical specialists. First of all, Dr. Emma Picasso, a pediatric endocrinologist, and Dr. Jacqueline Ansures, a nephrologist, who offer us a valuable insight into the diabetic nephropathy. They recognize that our technology could revolutionize and emphasize the treatment of diabetic nephropathy patients, and provide valuable insights into the fundamental backbone of our proposal, the biomarker of interest for detection. Based on their feedback, we began making critical decisions about the biomarkers that we were trying to use as a detection system. But our mission goes beyond research. We realized the importance of educating community about diabetes mellitus and nephropathy. On October 20 of the present year, at the Tecnológico de Monterrey Campus Estado de México, we brought our vision to the life with Sensibilización e Información de Diabetes Mellitus, a campaign that was a key part of our initiative with help of the organization Con Diabetes y Se Puede. This collaboration allows us to gather valuable information and receive suggestions on innovative ways that we can reach the needs of these patients.
The development of Nephro Prot considered local and international standards. In Mexico, it followed Mexican safety standards for diabetes mellitus prevention and treatment, shown here. Internationally, it complied with FDA regulations, including regulation of laboratory development tests, LDTs. For test safety and efficacy, it is also utilized ISO 9001 of 2015. For quality management, these regulations collectively ensure the safety and quality of Nephro Prot throughout its production and use.